All right, A&P1 students, in this video we're going to finish up talking about a little bit more information on osmosis. So in the last video we talked about what osmosis was and why it happens, or I guess like why water moves the way that it moves between the cells and the area outside the cells. But in this video we're going to talk about something called tonicity. And so I'm going to go to our objectives real quick here. There we go. And the tonicity, the definition of it is just the ability of a solution to change the volume or pressure of a cell by osmosis. So we'll explain that a little bit more in detail. And so one thing that we should be familiar with is when we say a solution, we're talking about the solution. And in the case with the body, the human body, it's a water solution. And that's what our cells are exposed to, is different types of solutions. So remember in our last video, we talked about how if you eat a bunch of chips and you get a lot more salt in your bloodstream, that salt that's in the bloodstream is outside the cells, and that's going to cause water to want to move between the cells and the bloodstream, and water in that case would move out of the cells and get into the bloodstream to try to equalize the solute concentration on either side of that plasma membrane. So that's what, when we say a solution, we're just talking about that watery fluid that's outside of our cells. Okay, but our cells are really important. Remember, they're the smallest living units within the body, and they are the ones that perform various chemical reactions and make all of our body functions happen. So they're really important. So we need to understand what would happen to them when osmosis happens. So again, what's going to change within them, and their volume would change and the pressure within them would change if osmosis happened. So I'm going to go back to our textbook so we can look at some pictures here about osmosis and tonicity. Okay, so like I said, whenever the water crosses the plasma membrane by osmosis, the cell is going to either gain more water or lose more water. And again, it just depends on the solution that the cell is in or being exposed to. So, or what that solute concentration is outside the cell. So it's going to change. So I'm going to show you how the body maintains normal solute concentrations inside and outside the cells. And when it does that, that will allow the cells to pretty much maintain their normal volume and pressure, and so they don't change much as far as shape. So they maintain their normal shape, and remember, if they maintain their normal shape, then they can function properly. So I'm going to go down to our pictures here, and what they, oops, I went a little too far. What they did was they showed, now they're showing just the red blood cell as an example of a cell in the human body. But realize this could be any cell in the human body. This could be a skin cell or a kidney cell, a muscle cell. It could be any of those. But in this case, they're using red blood cell as their example. So red blood cells float in the bloodstream, and so they have a plasma membrane. And there'll be a certain number of solutes. There should draw on the solutes as these purple structures. A certain number of solutes inside the cell, and a certain number outside the cell. And so what the body tries to do is to maintain an equal amount of solute concentration outside the cell versus inside the cell. So it will try to maintain that, and it can do it through osmosis. So in this case, the solute concentration, they're calling it interstitial fluid, but it's the fluid that's outside the cell, it's the same concentration as the solute concentration that's inside the cell, which they're calling cytosol. So inside the cell. So if we have that situation, water doesn't necessarily need to move because the solute concentration is the same outside the cell and inside the cell. And so water will still move across the plasma membrane. So you can see these arrows. But if, say, one water molecule moves across the plasma membrane, another one will come in. And then, say, 2 move across the plasma membrane, 2 will leave. So there's not really any net movement of water. You're not getting like a bunch of water leaving the cell or a bunch of water coming into the cell in this situation. So in the human body, the type of solution that our cells are in, we try to keep this solution where it's the solute concentration's the same outside the cell as inside the cell. Now, they call this, there's a term for this type of solution that this particular red blood cell is in, or any of our cells would be in. And they call it isotonic. So ISO means same, and then tonic is referring to that ability for a change in the volume or pressure of a cell due to osmosis. So when they're, and remember when we say isotonic, we're referring to the solute concentration being the same outside the cell as inside the cell. And let me find that word. Yeah, it's at the top here. So that's an isotonic solution. So our blood, since we're using that as our example, our blood is, and our red blood cells specifically, when they're floating in the bloodstream, they're in an isotonic solution, which means that that watery solution that's outside the cell has the same solute concentration as the solute concentration inside the cells. So water moves, but it moves in equal directions. So you don't have any sort of gaining of water by the cell or losing of water by the cell. So it pretty much stays the same. And then the red blood cell maintains its shape. So this is a red blood cell, and you can kind of see its shape. It's kind of more disc shaped, we call it. And then it has like this area in the center that's thinner. And so they call it biconcave because it's actually, got a little concave side on the other side too that we can't see. So that's a normal shape of a red blood cell. So we try to keep our red blood cells in an isotonic solution. So we have these homeostatic mechanisms in place that do that. So that way our red blood cells maintain their shape and then they can function properly. And their function is to carry oxygen. So as long as they have that shape, they can carry oxygen very, very well. And everything's good. All right. So this is what we need to be familiar with, other solutions that our cells could be exposed to. And the names that we call those type of solutions. So remember, iso means same. So this means the solute concentration outside of the cell was the same as the solute concentration inside the cell. So water did not, there's no net movement of water. It moved equally between those two areas. So the cell maintained its shape. So the next example of a solution is we may have a situation where the solute concentration, so the amount of solutes in this watery solution outside the cells or the interstitial fluid, is going to decrease. So we're going to lose solutes in that case. So that may happen with so if we'll use sodium and chloride as an example, but it could be other solutes too. But if we lose solutes, maybe through some sort of process like the loss of like a lot of solutes through sweating and things like that, what we'll get is this situation where our cells, in this case we're using red blood cells as an example, will be in a solution that is got less solute concentration than the solute concentration inside the cell. So we call that hypo. Remember, hypo means below. So that just means a low amount of solute concentration in this solution that's outside the cell. And remember that if we have solute concentration differences, then water is going to want to move by osmosis. So water will move across that plasma membrane. And it always wants to try to equalize the solute concentration. So if the solute concentration is decreased out here, then water is going to want to move away from that area. And so it'll move into the cells, and that'll help to concentrate the solute concentration, increase its solute concentration outside the cell. And then the water moving into the cell will help, because then it will be able to dilute the solute concentration that's inside the cell. And so that'll cause the equalizing of the solute concentration inside the cell and outside the cell. So in this case, if we put cells are exposed to a hypotonic solution, then our water is going to want to move into those cells. And so, but what can happen is if we have a pretty significant change in our solute concentration, which means like it really drops pretty low, we can have like a really rapid and sudden increase of water movement into our cells. And what will happen is they'll take on a lot more water, and they'll start to swell. And so you can kind of see that this one's becoming more bulgy and kind of losing its normal shape like it was over here. So you can see it's changing shape. So the problem with this is, like all of our body structures, if it loses its normal shape, the shape determines its function. So now this particular red blood cell can't function properly. So because of its abnormal shape at this time. And this could even, if it's significant enough and we have enough water moving into this cell could also rupture too. So it can just take on so much water so quickly that it damages the cell membrane or plasma membrane, and it ruptures, and then it's destroyed. And of course, then it definitely can't function at all at that point. So, so we want to kind of avoid these sort of massive changes in the solution that our cells are in. So we want to, our body has mechanisms, homeostatic mechanisms, to keep the solutions around our cells isotonic. So to avoid sort of changes in the shape of the cell and damage to the cells. But so this is stuff we need to be aware of and how to avoid it, which we'll do a case study in class that kind of talks about how we can kind of apply this concept to practical situations too. All right, and so because this can happen, the opposite can happen too, where we can have a situation where we get way more solute concentration. Solute increases drastically outside the cell in this watery solution. And so we kind of describe this watery solution that has a lot more solute concentration outside than inside the cell. We call it hypertonic. So hypo means like above. And so that just indicates that we have way more solute concentration in this solution outside the cell than compared to inside the cell. So remember, what will happen is the water is going to move by osmosis to try to equalize the solute concentration on either side of that plasma membrane. So how it will do that is water will leave the cell to try to concentrate the solute concentration inside here and increase it. And then it'll come out here and try to dilute this higher solute concentration out here. And it'll keep moving until that happens. Problem is, we can have a shriveling up of our red blood cells, or any cell in the body. So remember, that was our normal shape. And then if our cells are placed in a hypertonic solution, they'll do this little shriveling up situation. And again, wrong shape, just like this was the wrong shape, can't function properly, not going to be able to carry oxygen well. So, all right, so those are our 3 types of solutions our cells could be in. Remember, isotonic, our body has homeostatic mechanisms that keep our cells in an isotonic solution the majority of the time. So these are situations which could happen, and we need to be familiar with them, and we'll use a case study to kind of explain how you could maybe have these situations happen. Okay, I will see you guys in the next video.